Hello, I'm Nicholas Dowell. I work at the Clinical Imaging Sciences Centre, part of Brighton and Sussex Medical School, and these next few slides are an overview of my research. So I've done quite a bit of microstructural brain imaging, and some of the techniques I've learnt there I've applied to studying elsewhere in the body, such as the intravertebral disc. And the project I'm currently working on is looking at blood-brain barrier permeability and how to use MRI to study that. So all of these techniques involve quantitative MRI, which is using the MRI scanner as a measuring device. So using some of the quantitative MRI techniques on the screen here, we're able to extract biomarkers, which can tell us something about things that are happening in the body. So brain microstructural imaging has been applied to looking at aging, where I've identified differences between carriers of the ApoE4 gene, which is the best genetic risk factor for developing Alzheimer's disease. And I've shown there are particularly tract-specific differences that are found between those who carry the gene and those that do not. But also we've identified differences looking at inflammation as well, where we've been able to correlate changes in neurite density in this basal ganglia region with later fatigue for people being treated with the pro-inflammatory interferon. You can also use quantitative MRI and these microstructural imaging techniques to look at myelin and axonal density using techniques known as NODDI and McDespot. I've developed quantitative MRI biomarkers for the intravertebral disc. Using the T2 and T2 star value, I can calculate the variation in water content across the disc, and using magnetization transfer, this is a good marker for finding areas that are high in fibrous or macromolecular tissues, such as the outer edges of this intravertebral disc. Also, spectroscopy can be used to identify individual proteins. For example, here, this is a proteoglycan peak, which is a marker for disc hydration. We can also use quantitative MRI to study blood-brain barrier permeability. By injecting someone with gadolinium contrast agent, we can increase the signal intensity in areas of the brain where blood-brain barrier is compromised or there's high permeability, and this results in this increased signal intensity, which we can then model to calculate the extent of permeability. So my role has been to optimize all the acquisition parameters, understanding how the signal intensity changes with regard to acquisition parameters, and I've developed software to allow users to analyze the data and created fitting routines using C programming to fit the data to the model to extract the important parameters. And this has allowed us to produce permeability maps across the whole brain. It can be used in individual regions of interest, but also across the whole brain, as shown here. But this work is ongoing. As you can see, there are certain areas of the brain, especially in the cortex, which do not fit the model well. And so this requires further correction for artifacts and other drift within the scanner. These are my collaborators, and this is my contact details, and there's also a book that I've edited on many of these aspects too.